Hi, I'm Dan Levy, an NHLBI investigator and a BU faculty member. Framingham is the beneficiary of a large NHLBI program called TopMed that involves the sequencing of over 200,000 people. Remember that the first human genome was sequenced back in the year 2000 and cost $200 million for just one genome. This program that now involves Framingham includes 200,000 people with whole genome sequencing, many thousands of people with proteomics, metabolomics, DNA methylation, and more. And much of the work that I do in Framingham now involves the integration of these types of large-scale omics data, of which Framingham is a participant and great beneficiary. The overall aims of TopMed are shown here: to apply these transomics methods to identify molecular signatures of disease, to discover causal genes and pathways for disease, and to advance precision medicine. The work that I'm doing with my team and other members here in the audience today involves integrative transomics, where we integrate information from whole genome sequencing with information from DNA methylation profiling, information from RNA sequencing, and information from proteomics. And if you look at this triangle that combines those different omics resources together, if you look at the very top at genetic variation from whole genome sequencing, you can use it to connect to gene expression. You can use it to connect to DNA methylation or to proteins to identify the genetic variants that are responsible for all the variation that we see in our proteome and metabolome and transcriptome. Here's a series of 3 projects. We'd like to highlight some of the investigators here. Chun Yu at the top is a BU professor, Jin Tao, Amina Robi, all members of our research team, and a number of other people. We identified EQTLs, the genetic variants associated with changes or differences in expression of genes across the genome. And when you take whole genome sequencing in many thousands of people, 3 billion base pairs of DNA in each of those people, and look at the 20,000 genes that we have in our genome, that's a massive computational chore. Robi is a computational genius who was able to use cloud-based computing to put these resources together. We also were able to look at MQTLs, how genetic variation changes DNA methylation, and EQTMs, how DNA methylation changes gene expression. Another area we've been involved in is new, and that involves alternative splicing of our genes, allowing us to have greater diversity in our proteome than would be expected by our mere 20,000 genes. And one example of that is a gene called OAS1. Jennifer Huffman, from a former fellow in Framingham, was one of the people who identified a genetic variant in OAS1, identified that it was associated with alternative splicing, and was protective against severe COVID. On this slide, you could see our work looking at many thousands of Framingham participants, identifying 2 variants that are within the OAS1 gene, alter the splicing of that gene to create a protective form and a harmful form of OAS1, the harmful form associated with greater risk for COVID. And you can see the p-values are exceptionally strong, linking the genetic variant to the alternate splice form of OAS1 and linking that in turn to alterations in our white blood cell count. We believe that this kind of integrative work will help us understand how our genetic diversity results in risk for disease and also can predispose to a longer and healthier life. Thank you.